Hello everyone, welcome to Code School. So today we are going to discuss about the chapter Respiration In Plants. So in this chapter there are totally 7 topics.
Okay.
So here the first topic is do plants breathe?
We already saw the last chapter.
Okay. And then glycolysis,
Okay.
fermentation,
Okay.
aerobic respiration is our 4th topic. And under aerobic respiration there are 2 subtopics. The first subtopic is tricarboxylic acid cycle. And the second subtopic is electron transport system and oxidative phosphorylation. So the first one we are going to see about respiratory balance sheet, amphibolic pathway, respiratory quotient. Okay, so we are going to discuss about these topics in detail. So first we will discuss about do plants breathe?
Is it sheet or sheet?
Sheet. No, sheet.
Okay.
Not HU, not no. It is a
So sometimes you say sheet.
that is substance. Okay. So
Sheet
here
is a substance.
Respiratory balance sheet is that we are going to calculate the balancing how much amount of ATP and NADP is used in it. Okay. So we are going in detail about these 7 topics. So first we go with do plants breathe. Okay.
Okay.
So now we are coming to the first topic in respiration in plants, which is do plants breathe.
Okay.
So we know this already. We have seen in the last chapter itself in photosynthesis that plants breathe. So plants require O2 for respiration, oxygen for respiration, and they give out carbon dioxide. Okay. So for this respiration, plants do not have any respiratory organs like nose, lungs, and everything. They do not have any respiratory organs like we animals have.
They only have a stomatal.
Yeah, only through stomatal pores, lenticels, through that organs only they respirate. And each plant part takes care of its own gas exchange. Each plant part, okay. And very low amount of gas is needed. To only a small amount of gas travels from one cell to other cell. So very low amount, not how much gas we take in, how much oxygen we take in. So like the plants do not take. They only need a small amount. Okay. So here. So in the breakdown of glucose, okay,
C6H12O6.
yeah, C6H12O6. So in the complete combustion of glucose in the presence of oxygen, 6O2, it gives 6CO2 plus 6H2O plus energy. Okay. So it gives carbon dioxide, water, and energy. It gives out carbon dioxide, water, and energy. So this energy, it can be released as heat. Not all the energy is released as heat. So some heat is required, some energy is required to synthesize other molecules that is required, like rubber, latex and everything, whatever the molecules needed. So these cell prepare that, synthesize those molecules, okay.
Yeah.
So here, for that glucose should be oxidized. Glucose should be reacted with 6 molecules of oxygen to bring out energy. So glucose should be oxidized, right. So glucose is oxidized in several steps. So we have written here C6H12O6 plus 6CO2, it completes in one reaction. But for this reaction to happen, there occur several reactions. Okay, so oxidize in several steps. So this energy released is coupled into ATP synthesis. Okay, adenosine triphosphate. This energy group, it is bonded to ATP, and whenever needed, this ATP is used in plants. Okay. So in all living organisms, both anaerobic, aerobic, and all living organisms which do not live in oxygen conditions also, these organisms are known as anaerobic organisms. So all living organisms do partial oxidation of glucose. Okay. They partially oxidize glucose, okay, without the presence of oxygen. So this partial oxidation of glucose, which is glucose to pyruvic acid, is a partial oxidation. So this process is known as glycolysis. So all organisms undergo this glycolysis. Okay. So after this glycolysis only it is separated that anaerobic conditions and aerobic conditions. Okay. The first we are going to discuss about glycolysis in the next topic. Okay. Now we are going to see about glycolysis, which all organisms undergo this glycolysis, which is a partial oxidation of glucose. Okay. So here it was discovered by 3 scientists. Gustav Emden, Otto Meyerhoff, and J. Bernoulli. So therefore it is known as EMP pathway.
Okay.
Emden, Meyerhoff, and per mass. Okay. So it is also known as EMP pathway, this glycolysis, and it undergoes in both aerobic and anaerobic organisms. So in anaerobic organisms, this is the only process in respiration. Glycolysis is generally occurs in cytoplasm of the cell. So we will see what is glycolysis here. Okay. So first glucose. Okay. So glucose is converted into glucose 6-phosphate by ATP is converted to ADP. Okay.
Adenosine.
1 ATP is converted to 1 ADP.
Adenosine triphosphate to
Adenosine diphosphate. 1 phosphate is used and therefore it becomes glucose 6-phosphate. Okay. So and this glucose 6-phosphate is differentiated into fructose 6-phosphate. Okay. And fructose 6-phosphate is fructose 6-phosphate produces fructose 1,6-biphosphate by again the use of ATP
adenosine triphosphate
and it is 1 ATP to ADP. So this fructose 1,6-biphosphate, it becomes triose phosphate, 2 into triose phosphate. There is various triose phosphates. So this one is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, it is divided into 2 and each reaction yields the pyruvic acid. Okay, it is divided into 2. So from here it becomes 2 into 2 into 2 into 2 all the reactions. Okay, so 2 into triose phosphate. From 2 into triose phosphate it becomes 2 into triose biphosphate, which is known as 1,3-biphosphoglyceric acid. So with this it transfers to NADP plus to NADH. Okay, 2 NAD plus to NADH. So 2 molecules of NAD is transferred to 2 NADH. Okay. So after becoming 2 into triose biphosphate, it becomes 2 into triose phosphate. Okay, with 2 ADP transformed to 2 ATP. So here ATP to ADP. Here ADP to ATP. Okay, 2 ADP is transferred to 2 ATP.
Opposite ADP.
Yeah. Okay. And now this 2 into triose phosphate forms 2 into 2 phosphoglycerate.
Okay.
And this phosphoglycerate by the release of water molecule forms 2 into phosphoenol pyruvate PEP. Okay. So after that it becomes 2 into pyruvic acid, which is the end product in glycolysis with 2 ADP, 2 ATP. Okay. So here number of ATPs produced are totally how much?
2 ATP here and 2 ATP here. So that one and that one.
Can you say number of ATP produced, not utilized?
Okay.
So only 4 ATPs are produced from 2 into 2 ADP. Okay. So 4 ATP produced. Okay. So how many ADP produced?
ADP. Same 4.
No, how many ATP utilized? Utilized.
ATP utilized. ATP. Okay, that's for 6.
How come 6?
Here 2, there 1 and that one.
ATP utilized.
Only 2.
Yes, it is taking in ATP. Here it is ADP only is taken in. Okay. So ADP is only taken in these 2 reactions. Where ATP first understand the reaction clearly. Okay. Here 1 ATP is converted to ADP. 1 ATP to ADP. 2 ADP to ATP. 2 ATP. 2 ADP to 2 ATP. Okay. So here totally 4 ATPs are produced. Okay. By utilizing 4 ADP. There 2 ATP are utilized and produces 2 ADP. Okay. So here 2 ATP is utilized, so therefore minus 2 ATP. Okay, it is utilized. Here the bond is formed, the chemical bond is formed, there the chemical bond is utilized. Okay. So it becomes 2 ATP, totally production of ATP is 2 ATP. Okay. So in the middle there is 2 NAD plus which forms into 2 NADP, NADH. Okay. So 1 NADH is equal to 3 ATP. If 1 NADH produced is equal to 1 NADH is equal to 3 ATP. So for 2 NADH, how much ATPs are produced?
For 1 NADH, there are 3 ATP produced. For 2 NADH, there are only 6
ATPs produced. So it's equal to plus 6 ATP. So therefore the net gain of ATP is equal to 8 ATP. In glycolysis there is 8 ATP bonds formed. Okay, for one glucose molecule. Okay, can you understand this pathway? So these products are very important, and wherever the ATP produced are very important. Okay, so glucose to glucose 6-phosphate, 1 ATP is utilized, and fructose 6-phosphate. From fructose 6-phosphate to fructose 1,6-biphosphate, 1 ATP utilized. Okay. So and then 2 into triose phosphate, where dihydroxyacetyl phosphate and glyceraldehyde phosphate are formed. So from here 2 NAD plus is gives 2 NADH. Okay. So again 2 into triose biphosphate, it gives 2 into triose phosphate, 2 into 2 phosphoglycerate, 2 into phosphoenol pyruvate, and 2 pyruvic acid. So here 2 molecules of pyruvic acid are produced. Okay. Can you understand this glycolysis process? So next we will see now glycolysis takes place in both aerobic and anaerobic organisms. Okay. So aerobic organisms after glycolysis they continue another cycle called Krebs cycle, aerobic. Those organisms.
This is anaerobic cycle. That is aerobic cycle.
This occurs in all the organisms, both anaerobic and aerobic. Okay. So after this, the aerobic organisms continues to perform a Krebs cycle, and anaerobic organisms perform fermentation and alcoholic fermentation, etc. processes. Okay. So this process is common in both anaerobic and aerobic. Okay. So the end product of glycolysis is 2 into pyruvic acid. Okay. So we will go to the next one, which is fermentation, which is occurred in anaerobic organisms. So now we are going to see fermentation in anaerobic organisms. Okay. So this fermentation is carried out from pyruvic acid. Okay. Already it is oxidized to pyruvic acid. So from pyruvic acid it is divided into producing ethanol and carbon dioxide and producing lactic acid.
Okay.
So first we will see how some organisms by anaerobic respiration produce ethanol and carbon dioxide will be seen. So examples are yeast. So they produce ethanol and carbon dioxide. So yeast, the pyruvic acid, which we saw the last product in glycolysis, it by undergoing by the enzyme pyruvic acid decarboxylase and alcohol dehydrogenase, it forms ethanol and carbon dioxide. Okay, by NADH plus H plus releases NAD plus. Okay, and here in bacteria, okay, so in bacteria NAD plus is released from NADH. Okay, so in bacteria pyruvic acid is converted to lactic acid by the enzyme lactate dehydrogenase.
Okay.
So here NADH plus H plus is converted to NAD plus. Okay. So we have already seen that 1 NAD plus, the formation of 1 NADH plus H plus gives rise to 3 ATPs. Okay. So here if 1 glucose is oxidized, it gives 2 ATP. Okay. So here less than 7% of the energy in glucose is released here. Less than 7% of energy in glucose is released here. Okay. So here yeast poisons themselves. When the concentration of alcohol reaches up to 13%, so it should not exceed 13%.
It's like poison for them.
Yeah, it is. Yeah. Okay. So therefore, therefore many yeast conditions like bread and everything, the concentration will be less than 13%. If it is more than 13%, it will poison themselves. Okay. So this is the fermentation process. Here the main point is pyruvic acid, which is the end product of glycolysis. All the organisms undergo this pyruvic acid and produce pyruvic. All organisms undergo glycolysis and produce pyruvic acid. So this pyruvic acid, in case of fermentation, it is of two kinds. One is producing ethanol and CO2, like in yeast, and other one is producing lactic acid, like in bacteria. So we also produce lactic acid in our muscles in anaerobic conditions. Okay. So this ethanol plus CO2 is undergone by pyruvic acid decarboxylase and alcohol dehydrogenase. So there it is a lactate dehydrogenase enzyme. Okay. So for one glucose it produces 2 ATP as net ATP, and these are the things to remember in fermentation. Okay. So here only partial oxidation of glucose takes place in fermentation. Whereas in aerobic respiration, like in eukaryotes and like other animals, other organisms, which take in oxygen, so they undergo complete oxidation of glucose. They completely oxidize glucose and form organic substances, and they produce ATP bonds and everything. So now we will be seeing about aerobic respiration. Okay. And what are, after pyruvic acid, what it undergoes in aerobic respiration, we will be seeing. Okay. Mainly we are going to see Krebs cycle under aerobic respiration. We are going to discuss about aerobic respiration.
Okay.
So aerobic respiration is from pyruvic acid, which is formed from glycolysis. Okay, in anaerobic air respiration, glycolysis is transported to ethanol and carbon dioxide and lactic acid according to the organism. So in aerobic respiration, that pyruvic acid is converted into energy and ATP bonds and everything. So this pyruvic acid from cytoplasm, this glycolysis occurs in cytoplasm. So pyruvic acid is formed in cytoplasm. So this pyruvic acid have to be transported from cytoplasm to mitochondria in the cells. Okay. So it is transported to mitochondria because mitochondria is the energy house of the cell. It produces ATP. In the first process occurs in matrix of mitochondria, which occurs inside the matrix of mitochondria, and the second process occurs in the inner membrane of mitochondria, between the inner membrane of mitochondria. Okay. So after pyruvic acid, this pyruvic acid which is produced in the cytoplasm enters into the mitochondrial matrix. It undergoes oxidative decarboxylation. Okay. It undergoes decarboxylation oxidatively. So this oxidative decarboxylation is catalyzed by pyruvic dehydrogenase by this reaction. So 2-pyruvic acid plus acetate coenzyme A plus coenzyme A plus NAD plus in the presence of magnesium ion and enzyme pyruvate dehydrogenase, it produces acetate coenzyme A, the release of CO2 and 2 NADH plus H plus.
Okay.
So this acetate coenzyme A which is formed here, it again undergoes a cyclic pathway known as Krebs cycle. So it was discovered by Hans Krebs, so therefore it is known as Krebs cycle, and also it is known as tricarboxylic acid cycle. Okay.
Okay.
So next we will see about the Krebs cycle, which is tricarboxylic acid cycle. It is also known as citric acid cycle. Okay. So it is Krebs cycle, citric acid cycle, tricarboxylic acid cycle. Okay. So that cycle we will be seeing here.